Hi, I'm Richard Sever from Cold Spring Harbor Lab and BioArchive. With me, I have Sean Morrison from UT Southwestern. Sean, welcome to Cold Spring Harbor.
Thanks. Great to see you in 3 dimensions.
Yeah. Well, we were just talking about having met online, but not in person yet. You gave one of the introductory talks last night, which I have to say was really inspiring. And one of the things that really struck me was you talked really enthusiastically about the idea that we're in log phase for stem cell research, and that all these promises of cell therapy and gene therapy are finally being realized. Can you sort of expand a little bit on that optimism and why you think that's the case?
Yeah, part of the reason why I said that is that people have started to think about stem cell research as an old problem. You know, it was the frontier 20 years ago. But the reality is that 20 years ago, we were talking about how investment in stem cell research would lead to new cures and new insights into the biology that changed the way we thought about important scientific problems. And the field really is delivering on that promise. There's record numbers of applications to the Food and Drug Administration for new cell and gene therapies. Gene therapy is working. There are treatments that even go beyond what we hoped we would have at this point in the evolution of the field. And so the exciting thing is that the field has gone through that inflection point. And as you said, we're log phase in terms of developing new therapies, and there's so much new biology between the gene editing tools that we have, the new understanding that we have of tissue regeneration, and the capacity to apply these things to trying to cure the diseases that affect people.
And I think what's kind of amazing is that it's both you were talking then about the sort of translational impact, but also our understanding of the biology is growing. And one of the things you talked about was, there's many new factors that are being discovered, and new mechanisms, and sometimes there's this kind of sense that maybe we're just kind of like, looking in the lamplight and only kind of investigating the stuff we know about. But so there's new stem cell factors being identified all the time. Is that fair to say?
Absolutely. And if you look at where the new therapies are coming from, it all stems from basic curiosity-driven research. People couldn't have imagined in advance that there was a CRISPR to discover that would be incredibly useful for a lot of the stem cell populations that we're learning about now. We didn't know what the cells were or how they were regulated, and there's still so much to learn, particularly in the context of tissue regeneration, where everything changes relative to the steady state. And there are some tissues like the hematopoietic system where it's the same stem cells, whether it's steady state conditions or response to injury. But there are other tissues where the cells that are responsible for injury repair are different from the cells that are responsible for steady state tissue maintenance, or where there are different stem cell populations that repair different kinds of injuries.
Right. So, I mean, one of the things you talked about was your own very recent work in this area, and you were talking about regeneration within bone. So, paint the picture of that scenario. What is the when does the regeneration normally occur? What circumstances do we imagine this happen? And presumably that was your reason for looking at it.
My lab broadly studies stem cells and cancer. In the context of stem cells, we study both the intrinsic mechanisms within stem cells that allow them to self-renew and persist throughout life, as well as the extrinsic mechanisms by which the environment regulates stem cell function. For that, we focus on the bone marrow, where we identified the niche for hematopoietic stem cells. That's the specialized microenvironment where the stem cells reside and where they get the factors they require for maintenance. And the exciting thing that we've learned, particularly over the last 5 years, the key cell type that determines the niche are these leptin receptor positive stromal cells that we discovered and that line the outside of blood vessels throughout the bone marrow. And that's where the stem cells localize. The leptin receptor positive cells, when we first identified them, we figured they would be, we knew when we first discovered them that they were the key source of the known niche factors required for stem cell maintenance. But we figured that when we went beyond those factors, we would find other cells that were important in the bone marrow. And I'm not saying that other cells aren't important, but what has been surprising to us is that the more we study these cells, the more things that we find that they're doing. they're not just producing certain key growth factors, they're also protecting hematopoietic stem cells from the immune system. They promote vascular regeneration in the bone marrow. People don't think about it as much, but when you get chemotherapy, you're not just destroying hematopoietic cells, you also destroy the vasculature within the bone marrow, and that has to regenerate in order to regenerate hematopoiesis. So the story that I told last night was a new one where we've known, based on the work of Daniel Lucas and Paul Fournette, that there are nerve fibers in the bone marrow and that they're required to regenerate hematopoiesis after chemotherapy. But it wasn't clear, what cells those nerve fibers were interacting with or what the mechanism was by which they promote regeneration. And what we found is that it's the leptin receptor positive cells that regulate the innervation of the bone marrow because they're producing nerve growth factor that the nerves need, and in the absence of the nerve growth factor, the nerves disappear from the bone marrow.
So that must have been a surprise, the fact that, nerve growth factor, if you said to me nerve growth factor, I do not think bone marrow, and I certainly don't associate it with anything to do with leptin necessarily. That's kind of interesting. You think of all the stem cell factors that stem cell biology would think are coming out of stem cells. NGF is not one of them. Also, I have to say, nor would, leptin is not something that I think is that. What's the significance of the leptin?
Leptin, so the other thing that these leptin receptor positive cells do is they give rise to all of the adipocytes and osteoblasts that arise in adult bone marrow. We've heard about mesenchymal stem cells for decades and decades. Well, people for decades studied those cells by just growing cells from the bone marrow in adherent cultures. And it wasn't clear where the mesenchymal cells came from or what their physiological function is. But it's a subset of the leptin receptor positive cells that are those mesenchymal stem cells. And they give rise to the osteoblasts that are produced in the bone marrow that helps to maintain adult skeletal bone mass. And they give rise to all the adipocytes that accumulate in the bone marrow. And you asked the question about regeneration earlier. People had thought that the adipocytes in the bone marrow inhibited hematopoiesis, but at least for the subset that arises during adulthood, they're an emergency response system that helps promote the regeneration of the bone marrow after chemotherapy.
Okay. So you've got this interaction with the vasculature, you've got the interaction with the nerve cells. When NGF, comes out of a leptin positive cell and lands on a nerve, then why and what's happening then?
So that is necessary to maintain the nerve fibers in the bone marrow because nerve fibers require neurotrophic factors. So without that NGF from the leptin receptor positive cells, all the nerve fibers would disappear from the bone marrow. So that's back to your
maintaining the steady state of the niche, right?
And the way that the nerve fibers promote the regeneration of hematopoiesis after chemotherapy is that they secrete neurotransmitters that activate beta-adrenergic receptors on the leptin receptor positive cells and promote their ability to secrete multiple regeneration factors that regenerate hematopoiesis as well as blood vessels.
Oh, I see. Yeah, so again, that's another beta-adrenergic receptors. Again, not something one expects to hear in this kind of conversation. So that's creating a positive feedback loop then, and you get more NGF, and does that take you out of steady state? Is that the idea?
That's not important under steady state conditions, but it becomes important in the context of regeneration.
Right. So does that mean you get a sort of a temporary cycle just for the period of the regeneration?
Yeah, there's about a probably 3-week period that's really critical for the regeneration of hematopoiesis in the vasculature, and it's during that period that signaling from the nerve fibers becomes really important.
Oh, I see. Okay.
And this is a theme that we see across tissues, where nerve fibers promote the regeneration of lots of different tissues. And it's not just mammalian tissues, it's salamander limbs. There's a real evolutionarily conserved role for nerve fibers in regeneration. But in most cases, it was a phenomenon where if you cut the nerve fibers, you didn't get as good regeneration, but it wasn't exactly clear how it worked. And at least in the bone marrow, we're starting to figure out how it works.
And back to the sort of translational impact of a lot of these findings, one of the things that you mentioned was, thinking about, beta-2 adrenergic receptors. There are people who take beta blockers for various conditions.
Yes.
So how does that play into thinking of, you know, the impact of these findings?
So our prediction would be that people who are taking beta blockers at the time of a bone marrow transplant or at the time of chemotherapy wouldn't regenerate hematopoiesis as well. And we're in the process of testing that in mice. And if we see that in mice, then we'll go into clinical records and see if it is an unnoticed correlation.
Okay, yeah, that's what I was going to say. Presumably, I mean, you can do the experiments in mouse models, but the data must be there for you to go and look and see if there's a difference in outcomes.
Yeah, and the flip may also be true, where if we have agonists of beta adrenergic receptors, we might be able to accelerate regeneration. And this all goes back to that larger theme that we started with, where the biology that we're able to do now is so sophisticated relative to what we could do 20 years ago in terms of stem cell markers, in terms of the identity of the cells that create the niche, that we're able to look with more granularity at the mechanisms that regulate tissue regeneration. So I think this is going to accelerate our ability to discover things that enhance tissue regeneration. And it's for that reason that it's a particularly exciting time.
Yeah, well, it certainly seems like it, and you know, what a fantastic advert for blue sky research. It's been fantastic talking with you, Sean. Thanks very much.
Yeah, thank you.